drug discovery development, sir, heartily.
पण तुमचा बॅकड्रॉप तर फिनिशिंग स्कूलचा नाही आहे.
नाही आम्ही आता अजून फिनिश्ड होण्याच्या मार्गावर आहे आम्ही आता.
पण तुमच्या बॅकड्रॉपवर सॅन फ्रान्सिस्को कशाला?
आम्हाला वाटलं तुम्ही युएसए मध्ये आहे म्हणून आम्ही सॅन फ्रान्सिस्को घेतला.
नाही मी मुद्दाम त्यासाठीच घेतला होता. मी 1 जूनला इथे
हॅलो,
मी 1 जूनला इथे येणारच होतो. म्हणलं इथंच राहिलं बरं.
या सर. Dr. Satish, good to start now.
Yes, sir. Bobade sir will start the session. Bobade sir, can you just?
Yes.
Yeah, give me 1 minute. I will speak about Dr. Jamkar sir. And then
Sure, sir.
Yes.
10:30 ला आहे ना?
हो सर, झालं टायमिंग झाला
It's almost 10:36 sir now.
10:36. Yeah.
ओके.
Yeah. Please, Bobade sir.
माझा लॅपटॉप अमेरिकन दाखवतो.
Okay. So I, Dr. Chandrashekhar, welcome today's eminent speaker of this session, the recent trends in drug development. I, on behalf of School of Pharmacy and MIT-WPU, welcome today's expert speaker, Dr. Arun Jamkar sir, our constant support and force of motivation, our own Provisy, Dr. Milind Pandey sir, my colleagues, faculty, and aspirant student friends. Recently, this is a great initiative which is started by the School of Pharmacy, and I am witnessing from last 10 days, it has been remarkable to upgrading the knowledge of the students. On this same note, today's discussion is on recent trends in a drug development. Yes, the word has right, the recent, as medical history is always never a past because it always gone on a recent. And we are observing this how medical history is emerging in a great way. These drug development, starting from the pre-formulation, identification of new chemical entity, to the clinical trials, to the formulation and development, and up to the phase 4 is a tremendous process to bring a single molecule and to relieve the pain of the community. On the same note, we have invited today's eminent speaker, Dr. Arun Jamkar. So I request, with this introductory remark, I request Dr. Satish Pulshitiwar to offer online bouquet to our eminent today's speaker, Dr. Arun Jamkar sir. Welcome sir. On behalf of School of Pharmacy and MIT-WPU University, we welcome you.
Thank you.
Thank you.
You can send bouquet by Amazon.
So
yes sir, sure.
Before introduction of today's guest, I request respected Pandey sir to please speak his mind about today's program and about our today's expert speaker. Over to you, Dr. Pandey sir.
It's a great honor, my friends, to welcome and receive Honorable Dr. Arun Jamkar sir, the brain behind the transformation into the medical fraternity. He is the one who supported the integration of all the pathies, whether it be allopathy, Ayurvedic, homeopathy, Unani, even Ayush. So we are so lucky enough to have Dr. Jamkar sir with us today. As rightly mentioned, the former Vice Chancellor of the Maharashtra University of Health Sciences, then the former Dean of Surgery at the BJ Medical College, and on the top of it, he is a doctor, but he is the technocrat. He knows all the technology. He whenever you meet him, he always speak to you about the AI, then AR, VR, artificial intelligence, augmented reality, IoT. So I would like to tell you that more than an engineer, he is possessing all the technical knowledge as far as IoT is concerned, as far as AI is concerned, and not about the knowledge how it can be applicable for the overall development of the society, for the holistic health society. Dr. Jamkar is also working as a director at Persistent Systems Limited, getting into the various hackathons. The hackathon is he is the one of the pioneer member of launching a hackathon in India, and he is a constant supporter of it. He is working with the students on many more problem statement and guiding them, whether it may be a healthcare technology or the application of technology in healthcare. He is also associated with the MIT group as an advisor for the research and development program of the medical schools under the MIT group of institutions. I got a chance, or rather all of us, we got a chance to work with him very closely when
Yes.
we did our World Health Parliament. Let me tell you, he is one of the important pillar of the Parliament, and Parliament could see a great success under the leadership of Dr. Arun Jamkar. We students, we are fortunate enough to have Dr. Jamkar with us, a huge knowledge base, which he will be transferring it today for the benefit of not only the pharmaceutical students, but the overall knowledge strength at MIT-WPU University. So with this, I will wish you a very happy learning ahead. And thank you once again. Welcome you.
Satish sir, am I audible?
Yeah, audible. Pandey sir.
Hello?
Yeah. Sir, we lost you some in between last sentence.
Okay, last sentence. So I said I would like to wish all the participants a happy learning. And it's a great opportunity for all of us to listen, to appreciate, to convert it into the technicality, whatever Dr. Jamkar would be enlightening us today. So I take this opportunity to welcome Dr. Jamkar again, and looking forward for many more interactions with Dr. Jamkar and mentoring to the students of MIT-WPU University, to the faculty of MIT-WPU University Pharmacy School. I would like to also thank Dr. Limaye for coming up with such a good concept of finishing school, and I'm also congratulating Dr. Satish and Dr. Chandrashekhar and all the faculties of MIT-WPU University School of Pharmacy for getting involved deeply in finishing school. So thank you for giving me a chance. I would like to hand over back to Dr. Bobade, and welcome you, Dr. Jamkar, again. Thank you so much.
Thank you.
Thank you so much, sir, for your words.
Yes,
Now we will move ahead in the session. But student, definitely when we invite a great eminent speaker, so reading a biodata is not a formality, but behind that there is a big motto that we need to inspire, but what the work and what significant contribution that individual has did for the medical fraternity. I'm very happy. Before going to the formal introduction, Jamkar sir, I'm proud that we both belong to the same school, that school alumni, and I have a great time that I spent with you during the World Health Parliament, and I learned a lot. I'm here before you to introduce today's our eminent speaker, Dr. Arun Jamkar. Dr. Arun Jamkar, former Vice Chancellor, Maharashtra University of Health Sciences, Nashik. Sir's journey started from school education with a dreaming to be a doctor, and his dream became a reality when he admitted into JMC Medical College at Aurangabad. After that, he spent 3 dedicated decades. After that, he spent 3 dedicated decades in teaching, surgery, and performing operation and surgery at places like Grant Medical College, BJ Medical College, Pune. Sir is a great teacher, and a great teacher never stops his learning, and he is a lifelong learning, and that is what evidence he has pursued his PhD in surgical oncology from National Virology, Pune. Sir work in the capacity of Dean of one of the great medical college of the nation, that is BJ Medical College. Dr. Jamkar's contribution is noteworthy in managing the swine and disaster management of German bakery bomb blast in our own city, Pune. Medical fraternity appreciate the efforts did by Dr. Jamkar, and we can say that this is a fortunate fruit that after being a dean of BJ Medical College, sir has served the highest medical curriculum and medical education, that he has secured the Vice Chancellor of Maharashtra University of Health Science, Nashik. Over the years, he is a significant contributing in a critical healthcare ecosystem for interdisciplinary research. As Pandey sir has rightly mentioned, sir is trying to connect Ayurveda, homeopathy, artificial intelligence, technology, and whatnot. And with this ecosystem, his objective is to relieve the pain of community and to cater the healthcare need of the public. Teaching of bioethics, the last point I definitely would like to mention, Sir's immense contribution as a teacher in the medical curriculum. Sir has a seed thought of teaching bioethics to medical curriculum was his seed thought at National Model Center of UNESCO and bioethics as a subject in the All State University Medical College. So with this brief, I will not say it as an introduction, I will say it as a contribution. With this introduction, I welcome once again on behalf of School of Pharmacy and MIT-WPU University. And I request respected Jamkar sir to take the charge and please guide us and our student. Over to you, sir.
Thank you, Dr. Bobade, and thank you, Dr. Milind Pandey. I will share the screen first.
So click on the video also when you are sharing the screen.
Okay.
Yeah.
Can you see?
Yes, sir. Now it is.
Yes, sir.
Yeah.
It's showing.
Yes, visible.
Yes.
Very good. So let me first thank MIT School of Pharmacy, the principal, Dr. Kuchekar, Vice Chancellor of MIT-WPU University, Dr. Milind Pandey, Dr. Bobade, and everybody concerned. I am extremely happy to contribute in the finishing school. I have little apprehension about calling it finishing school because, like doctors, pharmacists need to be a lifelong learner. But then I think this is meaning to whether they are the finishing touches for the knowledge of pharmacy. So I'm going to talk a little different about recent trends in drug development. Thanks for the introduction. I just want to add that I had very large affection and passion about immuno-oncology. So my dissertation topic was on immunotherapy of cancer with porcine lymph node cells. Then subsequently, I did my PhD on NK cell interferon system and first clinical trial of interferon in oral cancer. With this background of immuno-oncology, I was invited to join as a chief medical officer with an Index Technology, which is an American company based in Cupertino, where they are into NGS-based mechanism to match 500 genes and find out which monoclonal antibodies are suitable and how to participate in those clinical trials. So this being my background, I thought I will talk a little more on all these aspects. Today, I'm going to have multiple sensitizing points. And instead of having only one points to be discussed for 1 hour, I'm going to talk first on AI system because AI system has been my passion. And AI system had made a tremendous development in all sectors of life, especially in image handling, like say low-hanging fruits. AI system has been used in diagnosing retinopathy, skin diseases and everything. But then today I'm going to talk on AI system in drug development. Then I'm going to talk on monoclonal antibodies and how they are being used with an NGS technology. And then because my immunotherapy background, I'm going to talk a little more about immune checkpoint therapy, for which we got a Nobel Prize last year. And then I'm going to talk on say recent trends, how the AI systems and wearables and IoT are being used in clinical trials, and briefly touch about in silico medicine, and then how the world is going towards what is called as umbrella and basket clinical trials. And lastly, I will talk on our invention with persistent and MIMER about a drug dispensing ATM, which works on symptom-based algorithm, where the drug dispensing ATM could be of interest to pharmacists. So these are all various domain areas of AI system. It is used in almost everywhere: skin diseases, retinopathy, and scopies, angiography, and everywhere. But I'm going to just talk on AI system in drug delivery. Now, the maximum development has been taken place in AI system in image handling, and image handling requires a lot of what we call as technical know-how and a chip called as a GPU rather than other chips. So this chip is created by NVIDIA, and the NVIDIA stocks have gone 10 times. I visited NVIDIA. There is a huge mural called I Am AI, and they sold a t-shirt, so which I'm wearing here. So I'm just briefly introduce about what is AI. So AI is artificial intelligence, where computers making decisions. And then John McCarthy has a term that it's a machine with intelligent behavior, such as perception, reasoning, learning, and communication. And once you say AI, these are the three terms being used. And I just want to just sensitize. So what is AI? What is machine learning? And what is deep learning? So AI is any technique that enables computer to mimic human intelligence using logic, if-then rules, decision trees, and machine learning. And machine learning is a subset of abstruse statistical techniques that enables machine to improve tasks with experience, and the category includes deep learning. So in a machine learning, the moment you give a data, the machine learns from the data and then create calls, and through that it becomes perfect. And whereas a deep learning is a subset of machine learning, which compose of algorithms that permit software to train itself to perform tasks. Now, deep learning is important in speech and image recognition by exposing multi-layer neural network, because this somehow I'm going to talk. Now, if you see Google Photos, they classify you on faces, or then if you see the say Facebook. So everywhere they are using deep learning techniques to identify the people. So there are various examples. AI, blue or electronic game characters, machine learning, IBM Watson, which is what called as say assistance for diagnosing cancer, and somehow it was been withdrawn. And the deep learning, natural NSL, then AlphaGo, and then driving systems. So I'm just going to talk a little more about machine learning, because it is going to say how it provides a system's ability to automatically learn. And then while the machine learns, we have two types, what is called as unsupervised machine learning and supervised machine learning. So in a supervised machine learning, there are various types where the image is given, and then you develop into a characters of those images, and then it identifies any new image, you can classify, and then it gives a diagnosis. Whereas in unsupervised machine learning, the computer AI itself creates the patterns of the images and then classifies them as the clusters. And therefore, these both things are important. So let us say we want to do machine learning in COVID pneumonia. So if you have supervised machine learning, then a radiologist will tell you that these are the 12 to 13 types of shadows in the lung. And then once you set in, you train the image handling software. Subsequently, any image of X-ray chest, it diagnoses almost with 90% accuracy. So this is a supervised learning. Now, sometimes what happens, we don't know that now COVID is new. So there might be a lot of other changes in the lung which are overlooked. So if you do an unsupervised machine learning, so what it does is, suppose let us say we have 1,000 patients which are say PCR positive, and then we show them all those X-rays, it will cluster and then find out These are the image changes in X-ray chest. So both the things are more important. And therefore, the type of learning will supervise, unsupervised, and then it could be reinforcement where you do supervise first and then unsupervised component can be added. So there are various algorithms, what is called as a PCA, and then commonest is clustering, Gaussian mixture clustering, and then supervised we have support SVM. All these are various things. I just want to sensitize because as a pharmacy student, you need to know about all these machine learning techniques. So the deep learning would application of this going ahead to find out these algorithms and deep neural network. And then we have a deep neural network that once we put an input of some component of image, and then it passes through the system, and then we have hidden layers and we have an outcome. So let us say we want to call A as A, then it goes through this area, and then suppose we want to have multiple things available, then we have multiple hidden layers. Sometimes we require 30 to 40 hidden layers. So all these features are extracted and then they are matched, and then you have output. So I'm not going to just talk much because we need to concentrate that all these technologies are available, they're available free. And all these platforms are available. The famous one is called as a TensorFlow, and then Keras or maybe CUDA. So these are available for download, and through that, we can create all image handling software. So going with this background, so there are three main paradigms: the symbolic, the statistical, and sub-symbolic, where it goes through all these components of knowledge, perception, communication, reasoning, and planning. So what is the aim of all this technology is to go into what is called as a precision medicine. So precision medicine is going ahead, an emerging approach to disease treatment and prevention that takes into account individual variability of genes. They're putting all the genes, environment, everything inside. And we are going what is called as a reactive medicine. And then we are coming with precision medicine too, where the genetic data of personalized medicine. So this we are traveling towards a personalized medicine. And as a pharmacist, we should know what is personalized medicine and precision medicine, because digital monitoring will go ahead and do a perfect personalized medicine. So now with recent past, there has been tremendous data explosion. And then we have a large number of patients coming with a genetic pool. We have all those pools going down. And then we need to use all these data explosion for better diagnosis. So machine learning technologies are expected to work. Now I'm going towards how we are going to use in drug development, that it will have a quicker, cheaper, and more effective drug discovery. Some are skeptical, but then they're doing a great job. A classical example, Pfizer is using IBM Watson to have immuno-oncology drugs. The Sanofi has designed an UK startup, Excentia, AI system for metabolic drugs. The Roche, a subsidiary of AI system, they are working on cancer treatments, and biotechnology company Berg is currently in phase 2 clinical trial involving advanced pancreatic cancer. So all these, say, 4 noted areas where AI system is being used for new drugs. Now, with this background, now we were exposed with a sudden challenge of COVID, and then we didn't know what to do. And then we had a lot of drugs available. Now the new drugs that those are coming will take a lot of time. But then till then, the pandemic will kill more people. So here, the AI system was used to find out what is repurposing of drug done. So there's one company called Benovalent.
One day, AI might be able to conjure up necessary vaccines on the spot or repurpose drugs to deal with new challenges. The latter being something benevolent AI in London is already leading the way on. This company uses AI to better understand the mechanism of disease, correlating data on illnesses, drugs, and outcomes, and providing more information than any human being could ever come up with. And in this case, a potential lead. What are we looking at here?
This is a demonstration of our algorithm processing documents in real time, reading the abstracts of scientific publications to do with coronavirus, and extracting relationships between key biological concepts that we really care about to carry out drug discovery.
And at this point, we're just a few months into an outbreak that could really, really grow. How meaningful do you think this data is so far?
There's a huge amount of information being published. We're not just pulling out coronavirus information, but we're merging it with a lot of existing publications on underlying biology. Our algorithms reason over biology at multiple layers, from the nanoworld of proteins interacting on our cells, through this maze of biological processes, up to human-defined definitions of diseases.
It's very early days for the process, but it is suggesting what might be worth further investigation.
Now, a lot of people were looking at virals, antivirals, and how they might potentially, treat the disease. We looked at it from a completely different way. We said, what are the other types of approved drugs that might inhibit the progression of that disease in the body? So we surfaced a number of drugs, and then we did some experimentation based on that and further research. And then we came up with this one drug that we think is best suited. It has both anti-inflammatory properties as well as the ability to stop what's called endocytosis, which is what enables the virus to enter the lungs, which is the most, potentially dangerous outcome of the coronavirus.
The company stresses this is only conjecture. And although the drug in question is approved for use in rheumatoid arthritis, clinical trials and full scientific evidence would be needed before it could even come close to being used for this purpose.
What we're trying to do is use this technology in the service of science.
One day.
So, briefly, AI has been able to screen billions of molecules for coronavirus treatment. And then they were able to find out that the company, which is called the Benevolent AI, they took only one afternoon to use company's whole knowledge base. And then they were finding out 2 human protein targets. That is AP2 associated protein kinase, AAK1, or SAC kinases. And then through that, they found out that there is one drug which is used in rheumatoid arthritis, with the side effects are benign, and that can be used, and that is called baricitinib. So baricitinib is entirely discovered by AI to find out that it will be useful. And obviously, it is what is called the repurposing of drugs. And then we can combine with lopinavir, and that is metabolized. So these are the papers available about baricitinib, and now baricitinib is recently available for use of corona. So what is important is now we need to do a lot of biological data mining, going through all the drugs, and then the repurposing of them is available. And therefore, the whole angle of AI system in drug development is towards finding out these drugs and how repurposing can be done. Now, the corona or COVID pandemic has created what is called as infodemic. And there is so much of development available, but then we need to take with a pinch of salt. And because all of these developments are not peer reviewed, and you can take this COVID-19 Research Explorer, which is from the Google, where all publications are available, including which is published the day before. There's something called as a COVID Scholar. So I'm just trying to give this to find out how it can be done. So with this background, how we can use AI system for further, it's called as in silico medicine. That in silico medicine, the every step of pharmaceutical research and development could be done, and therefore the drug trials could be brief, drug trial could be cheap. So now our mission is to accelerate drug discovery. So we want to find out what is lead ID, hit ID, and target ID, and validation. So in silico medicine achieved preclinical candidates with novel targets. So there are various things called the pandaomics, chemistry 42, but then the AI system is being used for clinical trials, what is called in clinico. So now the in silico medicine using a novel target for pulmonary fibrosis in 18 months. So healthcare system has to move from what is called as a reactive health to a proactive health. And here the AI system is going to help you, because in reactive health, we look after, say, when the patient is unwell. Whereas in proactive health, AI system and the data is captured continuously through medical grade variables. And we find out, give alarms to find out when the patient or the person will be sick. So the tests are conducted around the corner, and therefore the whole system of AI is traveling towards a reactive health to a proactive health. So with this background, so to briefly conclude, how AI system is being used in repurposing drug development and in silico medicine is all achieved through artificial intelligence. Now let us go to my next point of immuno-oncology, where I'm going to touch about monoclonal antibodies and NGS technology. Now, just briefly introduce myself. As a postgraduate dissertation in 1975 to 77, I had worked on immunosensitized porcine lymph node cells in carcinoma maxilla. Briefly, take a tumor and put in mesentery of a, say, porcine mesentery, and then the lymph nodes, they become enlarged, and these are anti-cancer properties, and delivering those lymph node cells through regional perfusion was my project. And I think this is all about what is called as a great-grandfather work of a monoclonal antibody available today. Subsequently, there were a lot of sludge available where I work on immunospecific xenogenic lymph node cell transfer factor, where I think this is all far beyond, because then subsequently there was immunoRNA, and then the monoclonal antibodies will come, and papers available. On the PhD side, I did my PhD on NK cell interferon system, and then we were trying to use the interferon in oral cancer because it works with what is called as viral-induced tumors. And then we were trying to find out how the immune system works in all these areas. So my guide for post-graduation was Dr. Ghosh, who was a visionary, and my guide for PhD was Dr. Ghosh, and guide for my MS General Surgery was Dr. Gharpuri. So with this background, I'm just going to talk on a monoclonal antibody, because what is called as a hybridoma. And this is one of the biggest thing to happen, that Kohler and Milestone were provided with the Nobel Prize for hybridoma technology. And then first monoclonal antibodies was licensed by FDA, that is OKT3. Now, in a general surgeon, I'm a transplant surgeon, and we're too happy to use OKT3 because that was reducing the chances completely. And then in a monoclonal antibody, we take an antigen and sensitize, and then we form an antibody forming cells, and then they combine those cells with a, say, myeloma cell line, and then both together form a hybridoma. And then what you call as the spleen cells are isolated, which are producing B cells, and then we fuse with the spleen cells with the myeloma cells, and then allow the unfused B cells to die. And the moment the is available with antigen, it creates a monoclonal antibody for only one type. And now there is so many monoclonal antibodies coming. Now, briefly, as a chief medical officer to Indus Technology, we have seen that there are 94 clinical trials going on in monoclonal antibodies in cancer. So it's very confusing. So when it is called OMAP, that is a MURI. We call it ZMAP, that is chimeric. When it is ZUMAP, that is humanized. And fully human, that's a UMAP. So these are the terminologies available. And therefore, a monoclonal antibody works that once get attached, they selectively directly approach. Now, because this study was in my mind, because it's one of the, what we call as the background review of literature being done for my PhD. And we are working on IL-2, IL-3, IL-4. And subsequently, when the corona came, then everybody was talking about a cytokine storm. And then cytokine storm is mediated through IL-6. And then the cytokine storm is because of IL-6. And then suppose we stop that IL-6, the cytokine storm will stop. And therefore, here we have selective antibodies available. And then this is how the tocilizumab came in picture. And then I am very proud that before tocilizumab was introduced, I wrote to say journals that cytokine storm can be avoided by tocilizumab and could be used in either in combination with the favipiravir or something. And I think subsequently people were using tocilizumab. And now the problem is tocilizumab is working towards reducing the IL-6 action. As a net result, once you do tocilizumab, the levels of IL-6, they increase, and therefore the cytokine storm is controlled. But it is not a treatment of corona at all. Now we got two, I think, monoclonal antibodies available for directly treating corona, and that is MITWPU and casirivimab, which are directly attaching to the spike proteins and creating a complete, what you call as total cure. And these are being tested. There are now 4 antibodies available. I think now this whole thing is available to you on your website. And I want all of you to find out, now this is the future of corona, that the moment it becomes available, it can be treated. And we are not behind. The antibody cocktail was used for treating a COVID patient, casirivimab and imidivimab, in say in a hospital in Medanta in Delhi. So with this background, that the monoclonal antibodies, how they are being used in corona and how they are being used for treating, say, corona proper and cytokine storm. So now I'm going to talk and go towards the cancer. Now cancer is a genetic disease. And then we can see that there are multiple genes available. We have what is called as EGFR inhibitor, cyclin-dependent kinase inhibitor, then CTLA, telomerase, then selective anti-inflammatory drugs, then we have EGF. And if you see, there are multiple, all of these genes available. And therefore, each cancer comes with these genes. And what is required is to find out the genes can be stopped. So there is a story that one patient was diagnosed to have a myelodysplastic syndrome. And it was like a leukemia, where 4 different regimes were used for leukemia with no cure. And then when genetic testing was done, it was found to be uveal melanoma. And then there was a specific monoclonal antibody available for uveal melanoma. And then the moment it was available, the patient was cured. So this is the success of MOB. And suddenly there were several successes available. And then whole American society, in the Western society, started pouring money in development of monoclonal antibodies. And I think what is required today as a pharmacist to find out all these monoclonal antibodies and how they are being taken place in development. And while this is going on, the whole concept of immunotherapy got a boost with a Nobel Prize. Then 2018 Nobel Prize was given to Tosuku Honjo and James Allison for what is called as immune checkpoint inhibitors. Now this is an entirely new type of immunotherapy, where what they are doing is they are finding out how to modulate negative immune regulation. That T cell, I hope everybody knows, is able to kill a tumor cell, but while it's trying to kill, stop. And that break, suppose we have a monoclonal antibody against that break, that is what is called as immune checkpoint therapy. And then there are the various combinations available. I just want to show you that how the immune checkpoint inhibitors are working. And this is a small video which gives you immune checkpoint therapy.
Cancer immunotherapy is an innovative therapeutic approach to treat cancer by mobilizing a patient's own immune system instead of targeting tumors. It can conceivably be effective against any type of cancer. Immune checkpoint inhibitors, a promising new class of cancer drugs, can unleash the cancer cell killing power of the immune system by blocking the hardwired inhibitory pathways that keep T cells in check. The effect can be enduring and long-term, allowing the body to respond rapidly should a tumor recur.
Immunotherapy offers the potential to complement the high-impact short-term benefit of molecularly targeted therapy with a potentially more durable response mechanism.
Our immune system provides surveillance activity to kill tumor cells in the body. When a T cell encounters a tumor-specific protein carried by an antigen-presenting cell, the tumor antigen can trigger antigen-specific T cell activation and expansion. These activated tumor antigen-specific T cells will eliminate the tumor cells. Checkpoint molecules help keep activated T cells under control. They can be stimulatory or inhibitory. Inhibitory checkpoint molecules provide a negative feedback mechanism, like a brake on a car, to attenuate the function of activated T cells and prevent an overreaction that could damage tissues and organs. Cancer cells can utilize the body's immune inhibitory checkpoint system to blunt T cell action and enable the tumor to develop. Programmed cell death protein 1, or PD1, is an example of an inhibitory immune checkpoint receptor. When PD-1 interacts with a ligand such as PD-L1 and PD-L2, which may be expressed on normal or tumor cells, activated T cells will be turned off, inhibiting T cell signaling. Some tumor cells express high levels of inhibitory immune checkpoint ligands, such as PD-L1, allowing them to avoid destruction by T cells. Inhibitory immune checkpoints can be blocked using monoclonal antibody-based drugs. Immune checkpoint blockade prevents the binding of specific ligands on tumor cells to their inhibitory immune checkpoint receptors on T cells. These powerful antibody drugs can significantly boost a patient's immune response to a tumor. They may also cause serious side effects, such as an autoimmune response in some people.
The FDA approved the first immune checkpoint inhibitor in 2011 to treat advanced metastatic melanoma. Subsequent clinical trials have shown that this approach holds promise in other advanced cancers, including lung, kidney, bladder, head and neck, and Hodgkin's lymphoma. Potential promise is to increase the proportion of patients with durable responses and to convert the non-responders to responders using combination treatment approaches.
PD-1 is just one example of an inhibitory immune checkpoint molecule with a proven role in cancer immunotherapy. Other inhibitory checkpoint proteins include CTLA-4, TIM3, and LAG3. Research is underway to better understand the underlying mechanisms of immune checkpoint networks, to discover new checkpoint receptors and ligands, and to develop novel monoclonal antibody drugs for cancer immunotherapy. Clinical studies are also evaluating promising combination treatments that include the best standard of care therapies with one or more immune checkpoint blockade drugs to improve patient outcomes and survival.
So the point of contention is we have several monoclonal antibodies available against the say biomarkers and genetic gene receptors, and then the say immune checkpoint therapy has opened a new vista, where now the classical is the anti-CTLA-4, which is a new field, and then the whole thing is traveling together, that you can have directly the monoclonal antibodies attacking the tumor cell. At the same time, there are other monoclonal antibodies which are attacking the inhibitors. So I think this whole thing is coming in picture, and then the now whole thing is, I'm not going to repeat, because ipilimumab and the Keytruda, there is one thing which is working as a immune checkpoint inhibitors, and then for this they got a Nobel Prize. At the same time, now what is required is using an NGS technology, what's called next generation sequencing. And therefore, the moment we have a next generation sequencing available, we are able to find out the genomic content of a tumor, and then the costs are going down. And now we have what is called as all these drugs are available to match the gene combination, and then each one is available to have which monoclonal antibody is available. So if you see the graph, so the chemotherapy, target immunotherapy, and then slowly it will go up, and then the targeted chemotherapy will have a peak in 2025. And the global genomic market is growing at a CAGR of 13.3%. And then this is not only working in US, but it's working everywhere. And under the index technology, we are going to launch all these, say, gene parameters in India, which is called as iTreat and iPredict. So these are the major players in this field. And then we have various genetic agents companies available in India. These are around 10, like Datar Genetics, Strand, Medigenome, where a tumor is given and they give 500 markers. So what you do is we want to find out all these are available. Now, the first thing that is called as EGFR, epidermal growth factor. And then we have several monoclonal antibodies available, KRAS. So this gives a spectrum of all these monoclonal antibodies available. So what we do now is something called as a foundation medicine, where the genomic sequence is done, and then we find out which monoclonal antibodies are available in the clinical trial. And then there might be some trials completed or some trials useful, and then whole thing is available on, say, American website of clinical trials. And 75.6% oncologists are using NGS to have a perfect monoclonal antibody for treatment. Now, with this background, now what is required is NGS technology has to go beyond what is called as a cells. And these circulating tumor cells, they are RNAs, and then you can find out that these can be characterized. And then if you use NGS for a circulating tumor cell, and then monoclonal antibody load will be very low, and it can be used either for diagnosis as well as for treatment planning. I'm very happy that one of the faculty of MIT Pharma called Dr. Jayan Khandare, who has a company called Actorius Innovations. I want to give him, big appreciate all his efforts, because he's the one who is working with the nanoparticles on circulating tumor cells and circulating DNA, and which can be used for, I discussed with him, and I said, why not use NGS technology to find out all these with a monoclonal antibody? Maybe he will be in future. So with this background, what I'm trying to tell you is NGS technology being used to match the tumor and then find out which are the monoclonal antibodies available and then give a perfect match. And this is how we are traveling from a person. Now, what is happening further is now we are going far more detail available that we are working towards two types of the clinical trials. Now, these are all available in US, what is called as umbrella clinical trial and basket clinical trial. So in oncology, we do all these trials and we design the master protocols. So in an umbrella clinical trial, test well-known drugs and then allows new drug to be tested and approved quickly, then clinical trial. So in an umbrella clinical trial, what is happening is one drug, say one type of cancer with various types of gene markers. And in one trial, we are able to do 4 to 5 clinical trials with the same control. So in an umbrella clinical trial, we are discussing about multiple types of different genetic mutation. In a one, say carcinoma breast, we have 5 to 6 types. And therefore, in one clinical trial, same the number of say what you call as controls are small, and this whole thing could be done. This is what is called as umbrella clinical trial. Other thing, what is called as a clinical trial. In a basket clinical trial, one genomic marker which is available in multiple malignancies. So a simultaneous clinical trial is being done. Now this is about umbrella, where we have a target, say drug A, drug B, drug C, and in one particular clinical trial, multiple drugs are being tested. And then we monitor like everything. But then the advantage of umbrella clinical trial is simultaneously 5 to 6 type of drugs can be done. And this is all protocols of umbrella trial design. Whereas in a basket clinical trial, what we are doing is single disease. Let us say we are doing the basket clinical trial. So EGFR. So let us say EGFR. So anti-EGFR antibodies being used, and then lung cancer, colorectal cancer, ovarian cancer, multiple myeloma, breast cancer, all these are used for a single drug. I hope you all know about Herceptin. That when the Herceptin is for carcinoma breast, which was the first drug where there was a tremendous effect and efficacy was seen. As an end result, before the trial, the result was on, it was evaluated to use, say, Herceptin in carcinoma breast. So in a basket clinical trial, the same Herceptin can be used in all these types because the EGFR is available in all types of epithelial tumors. So I'm not going to much detail because this is a National Cancer Institute stream. And then we do, say, one drug with multiple cancers. Here I finish my third part about monoclonal antibodies and how we are conducting a new types of clinical trial called umbrella clinical trial. Now, let us go and find out how we are doing clinical trials with monitoring of IoTs and wearables. So I just want to give you my background that during that immunotherapy, I was trying to use regional cyclophosphamide chemotherapy. This I'm talking about 1975 to 77, where the cyclophosphamide is injected through carotid through maxillary. Those days there were no what you called as pressure injectors. The only way that we had was to increase the height of the bottle, and then this was done as a postgraduate thesis. And now this was what is called as a negative result that I did 16 cases and nothing happened to anybody except one patient where there was say 25% regression, and there are a lot of complications available. So this I declare as one of my biggest mistake of me, that we try to do a trial without doing a review of literature, because we were say getting biased that if you do a regional cyclophosphamide perfusion, the high dose would be available into the tumor site and it will regress. But then now cyclophosphamide, as you all know, requires an hepatic activation, and therefore there is no matter what happens you give regionally, it has to go to the liver to get active molecule. As a net result, it will fail. So this is something which gives a message that before doing all these trials and say drug therapies, we should have a complete review of literature. Now, I had an experience of doing several clinical trials. I had two types of trials. One was with postoperative pain and then bowel clearance. Then last three are very particular because I'm an urologist. I did say first clinical trial of India about Viagra, that is sildenafil. And it is entirely new thing because something, the sex is a taboo in India. Therefore, all these checklist and follow-up and all these was so confusing, rather so confidential. But then we were able to do the first clinical trial of Viagra before it was introduced. And then subsequently I did for tadalafil. All these both of things, they require erection to be there so that it can be sustained. Whereas there is another drug called apomorphine, where if you give say sublingually, it can create erection, and therefore I can create trials. So they are some of my trials. So now Internet of Things are coming in big way, and these are the network of devices. And now IoT is exploding and fast, and therefore we are using all these IoT for clinical trials. Now there are the various IoTs available, cloths and monitors and ECG monitors, and then we have what is called the variables. So whole thing is coming in big way, and we can find out that clinical trials of tomorrow, they will have all these like cuffs, wristband, breathomics, clothings, patches, so that once you give a drug, you want to monitor the toxicity, monitor the effects, you could do directly with all these variables. And these are coming in big way. And I think before treatment, the selection can be done with the variable. During treatment, it is improved evaluation. And after treatment, you can all monitor. And all these put together, the variable can give you better results and dogmatic results early. The classical thing, I'm just giving you some variables where skin provides an washable clothes and it can find out ECG level, sleep quality, and MIDI biosense. It's just trying to give you every monitor of cardiac thing on the respiration, live ECG, and these are all variables. And then we have called as respiraise, where it is giving you acute respiratory monitor. So all these variables can be used to find out various diseases, how do you progress. Now, as a consultant to persistent, we are trying to do a total knee replacement. And then new knees, those are coming. They are very advanced, what is called as a personalized knees. And they develop with what is called the 3D printing, so that you have an MRI available of the knee joint, and the selective personalized device can be created. Now this device is porous. As a net result, the Haversian canal system gets connected, and then with Haversian canal, the bone growing inside the prosthesis. So after some time, the prosthesis become part of the bone. These are all new things coming. And then how do you find out all these? Now they are using variables, that we are putting variable in the prosthesis. And then with the Apple Watch, they can monitor. And then they are giving a lot of role coming into the variables. And what is now required is now, so suppose you have a variable available, then you are able to monitor constantly round the clock the movement of say T velocity. And now this is a T-belt tray, how it is moving. And therefore we have in silico medicine coming in the medical device industry. So I think this is one which is more important, that a total knee replacement with a wearable can monitor the health of the, say, joint around the clock. And now we are using about expanding roles of simulation, and then simulation technology is being used for what you call as all medical gadgets, say the meniscus, cartilages, and therefore simulation technology is being used. And then we suggested that all these movements could be monitored. And then we have an European Commission publishing about all these health innovations, and it's called Avicenna Alliance, where the tibial movement can be used. So with this background about, I think I talk about wearables and IoTs and how they are being used in clinical trials, and those wearables and implantables can be used for monitoring total knee replacements, and we're giving the perfect results. And this is an extension of in silico medicine going towards all these prostheses. Lastly, I'm just going to talk about iDoctor, which we developed in Persistent. So iDoctor is an intelligent drug dispensing ATM, which based on a symptom-based algorithm. You can see here the two parts. The gentleman is sitting here, and then there is a drug dispensing ATM. So the theme is the moment you sit on that iDoctor chair, it measures your weight, it gives you fat analysis, then measures blood pressure, weight, BMI, ECG, pulse. So all these are recorded, and then the machine asks you about symptoms, and then you go down, and then the machine too many, say what is called questions, because it is going to just diagnose you based on say symptoms. And therefore, suppose you say vomiting, it asks you about the red flags where it is connecting to you, and so this is about how the patient interaction, it is Aadhaar enabled. Then we have intelligent diagnosis based on these AI-based system for algorithms, and then medical dispensing. And then it measures all these blood pressure, PO2, thermometer, body fat, everything together. Now, and then these are the kiosks available. Then we have patient interaction, diagnosis, and dispensing. We restricted to 50 clinical conditions, 50 diagnosis, and 50 drugs. And it's available in multilingual. So if you go in detail, it registers. And then what we plan to do is you want to set up in India, where the patient first time will be evaluated by a doctor, and the whole data is available in the system. And then when suppose somebody has a new symptom or a new operation, it has to be evaluated, and then the whole thing is ready. When the patient goes again with the user login, then your safety parameters, like as I said, and then the symptoms are asked, then the red flags, then we confirm the body system, we follow diagnosis system, and then we give them a drug. This is a detailed workflow that the moment the system says that it is not my cup of tea, then it just directly calls ambulance, and then this goes into whole cycle of an AI system based diagnosis. So this is how we are trying to do an iDoctor and develop those technologies available. And had a huge team. This was between MIMER Medical College and Persistent Systems, and where we did all these. And then the feature of medicine is you can talk over the symptoms on the phone, and then the iDoctor robo can deliver the drug. So this was about an iDoctor, where a drug dispensing ATM is one which is might be interested to the pharmacist. And this whole thing is available because while the drug is being dispensed, the scanner scans the say expiry date, and therefore it won't give the drug unless it is within the valid period. And then it has a whole mechanism by which, suppose you have 50 drugs, and then that whole inventory control is done through a cloud-based system so that the pharmacist knows which drug has to be replaced. So this whole thing is there. So with this background, I think I thank again for giving me chance, and I try to touch to the multiple points. And I just want to sensitize because MSc pharmacy students have a lot of scope in clinical trials, maybe monoclonal antibody trials, and then NGS technology trials. So I think the future is very bright. I just came yesterday from US. I think the pharmacists play a bigger role in US by dispensing drugs, because at the end, everything goes to a CVS pharma. And you go there, and then the whole drugs are dispensed through in a what you call as a specific bottles, and pharmacists play a bigger role. Thank you very much.
Thank you so much, sir. Indeed, as we heard from you during World Health Parliament, you talk about healthcare ecosystem, and with this webinar and presentation, we are really came to know how you are trying to connect, and in fact, you have connected almost all the points with the different disciplines, and you involve the technology. This is a detailed insights on technology, medical, pharmacy to our students. So we thank you on behalf of MIT World Peace University and School of Pharmacy for this detailed insights on this topic. Let me move ahead, sir. I request all the delegate students, sir, these are the empowerment postgraduate students from the pharmaceutical chemistry and quality assurance discipline, and even in fact few of undergraduate students are attending by their interest here. So student, I request you with the connection of all that sir has discussed, if you're curious to know something, if you have something to share and discuss, and you know more than that, so I request that we will start with the question answer session. So I request the student to unmute and please ask the student, ask the question to the sir.
Yes, the session is open for question and answer.
Yes, sir.
Yeah. So we have the very great personality that he is, sir is very much, experts in AI technology, in medical health, all these things. Probably it is, I think we have got very good opportunity to interact with the Arun Jamkar sir. Okay. So please students, feel free to ask any questions to sir, please. Before asking the students questions, so how to you, sir. Sir, you talked about the artificial intelligence, machine learning, deep learning, that we are moving from the drug repurposing to drug reformulations. We are moving from the full to penless drug delivery. So what is the role of, sir, 5G technology in all this? Because without that technology and faster communications, we cannot achieve all this success. So what would be your comments on this, sir?
I think 5G technology is going to, what you call as revamp whole healthcare system. First thing, let us say we are talking about variables. Now,
Yes.
data is big. And therefore, suppose you want to transmit that data from the patient to the cloud, you require a 5G. Now, I didn't want to complicate, but then we are working at present system what is called a smart ambulance, where all these AI-based data can be used, that a patient is picked up home, then we have a platform which connects with the EMR system, and then that whole ambulance becomes extension counter of a hospital. And because this requires a 5G, and therefore all the gadgets are available, all these variables are available, and then you can have ECG monitoring, you can do oxygen therapy, everything. As a net result, complications would be reduced. That now what happens, ambulance at present are just they carry patients.
Yes, sir.
They don't do anything, might be giving some CPR or something. And whereas now with a smart ambulance, the platform is available through the cloud, where you require 5G technology. The connecting doctor of emergency medicine will constantly supervise you. And then let us say somebody has a myocardial infarct, and then we have say TROP-T or some say investigations available, where a streptokinase can be given in the ambulance, so that whole thing can reduce mortality. Suppose somebody is coming with stroke. So suppose we diagnose stroke and all those investigations are available with connecting with the EMR of the what you call as receiving hospital and sending hospital. That whole thing can be managed, and then you can reduce the death rate as well as complication rate. So this whole thing requires a 5G. And we are trying to do with Apollo hospitals, and we might launch a what you call as a first smart ambulance in Hyderabad because they have 5G technology. But believe me, 5G technology is going to change whole healthcare. Now, even if you want to give CPR or you want to give all the machines are available which can work through 5G, and suppose somebody requires another set of instrument that can be sent by drone. In US, everything comes by drone, and it comes with all technology available, where the receiving hospital trains the bystander to take care. So I think 5G is going to be a milestone in the development of medicine.
Yes, thank you, sir. Thank you for your kind words.
Sir, again, as I discussed with you, I think, yes, Internet of Things and artificial intelligence is really a breakthrough innovations and will change the entire medical treatment. But apart from this, I have one question, sir. From last 1 month, it's troubling me a lot. That COVID-19 or this kind of infection basically hits to local areas of the body. That we are saying it basically affects your lungs. So my question, maybe I'm curious, maybe to know that it's infecting your nose, throat, or rather lungs. So instead of coming with a vaccine and injections, so can innovation think about to treat it locally, like a rota caps? Even I have heard or read that the nasal vaccine is on the way to come into. So is it possible in the drug development to convert the vaccine in the form of administrable form, patient compliance, and easy access and industry production? It will be great, sir, if you solve my query.
I think the best thing that can happen now, we are coming with a monoclonal antibodies for specific for corona, which can attach on the spike proteins. Let us say somebody has small symptoms. They'll spray, and then the increased concentration in a small amount can be done in the nasal mucosa. And I think the entry while the virus is inside can be blocked. So this is one thing. But now the problem is it is changing patterns so much. As I said just now, that we had never given importance to healthcare. Western societies are doing same thing. They were just spending billions of dollars on nuclear deterrence. And they created barracks and maybe say they create whole thing that even people can stay underground. And nobody thought that the next wars are not with nuclear bombs. They are with biological warfare. And say corona, I have a strong belief that it is a biological warfare which is created by China. And because you can't have some virus which works in Dubai, which works in Stockholm, which works in India, which works in summer, which works in winter, which works everywhere. How you can have a natural virus to do it that way? So this is one thing. And believe me, unless and until we create a system to handle such pandemics, what is best has happened is AI has come from the beginning. AI, I didn't go into much detail, but AI helped us for epidemiology. The moment the Wuhan virus came, then there was one gentleman who gave with a statistics of people traveling, exact dates on which it will come to Seoul, it will come to Stockholm, it will come everywhere. So AI is being used everywhere. And now with repurposing drugs, now I haven't talked of viral vaccine. That spike protein viral vaccine is an AI. Now, which component of spike protein has to be used for creating vaccine is again done by AI. So I think we need to prepare ourselves in such a way.
Yes.
Tomorrow, some other bad vaccine comes, or bad virus comes, or some other virus comes. We need to prepare ourselves. We are fast that within 3 to 4 months, we are able to create a vaccine. I think this is one of the fastest development that science can do. At the end, all warfares are to be handled by science. So we have to need to respect science and develop science. If you see all of us, we are paid peanuts, and everybody economist. Maybe somebody who is working in somewhere, they are paid in, say, millions. And therefore, the priority of the society need to change to respect healthcare, technology, and technicians. I think this is a message that we need to find out that how the whole priority of the society has to change to improve the healthcare. Now, I think spend at least 5 to 10% of GDP on healthcare. But even the Western societies, where they are spending around 14%, they collapsed. In National Health Service of UK, it collapsed. So I think we need to find out that whole priorities of healthcare has to be changed. I think that is the biggest message that the corona gave to us.
I think, sir, this AI technology, machine learning, deep learning, IoT of health things probably would be one of the best complements and supplement to the health fraternity. So in future, already we have been proven in pandemics. So you rightly said. So really, we are also moving on that, how we can integrate informatics, pharmacoinformatics, cheminformatics to introduce and integrate in drug discovery and drug delivery in order to accelerate the process. That you are rightly said, sir. Really, we are very thankful for that. Yes, Bobade sir, please, you can take it with other.
Yes, I think students, if you have any kind of a question, query, please go ahead, because this is a great opportunity for you all to extend your knowledge and thoughts related to the interdisciplinary. If you are an expert in a computer, please go ahead and seek the guidance from the...
Sir, Bobade sir, we have the MSc clinical pharmacy student, medical information student from Shivaji University also, Nitin Naik. Yeah, Naik, do you have any questions? He's from Shivaji University. He's from medical fraternity. Yeah. Please, Nitin, if you have any questions,
please go ahead, please.
Hello, sir. Okay, so I have a very simple question. Does everyone is wearing now their mask? Okay, sir. So does that wearing a mask habit, the oxygen means oxygen will take and we expel the carbon dioxide, and with that mask, it is possible that a lot of carbon dioxide again we should inhale. So does that affect our lungs and other respiratory systems?
No. Once you exhale, that carbon dioxide won't come again, because N95 mask or any mask is porous, and it has one way. And whatever you breathe out, it goes out. It won't come in. Now you are a student of bioinformatics.
Yes, sir.
So there is big thing happening in bioinformatics. Persistent, we also develop a bioinformatics protocol that all those viral vaccines with mutation, they could be monitored with bioinformatics. And the strains are available, and we could do an in silico medicine to find out which vaccine need to be upgraded. So all those mutation happening, now we have, say, South African variant and British variant and whatnot. So we can monitor them constantly. Now what is happening nowadays is technology is being used fast about viral replication and how the mutation is taking place. But then a bioinformatics has to work on what are the various modifications available. Now we gave this to Serum Institute as well as to NIV, how the bioinformatics protocol can be monitoring to create a vaccine to include all the mutation. And I think once one vaccine is available through the phase 3 trials, all these modifications can be done. But then the whole background material is required through the bioinformatics. I can share you our data of how we are using, say, all those genome sequencing to find out what are the modifications available, because that is another half an hour talk on how the bioinformatics can be used for improving viral vaccines.
Okay, thank you, sir. So we have also worked on that, and we have studied on few drugs, and we have just recently published one paper in medicine on coronavirus. So 3 drugs we have tested on the viruses, coronaviruses, sir, at Shivaji University under the guidance of Professor K.D. Sonavane, sir, who is a postdoc fellow from NIH Bethesda, sir.
Shivaji is a nice university. I had been there in campus. I was dean of CPR for 1 year.
Okay, okay. Nice to hear you, sir.
I was there.
Okay, I think, sir, thank you, Nitinji, for your question. Move ahead.
We'll take live feedback.
So
Please.
I request one of the any representatives to student, please come virtually here and please share what exactly.
Amruta T. Kadam, you can take the name. Amruta, are you there?
Okay.
Amruta T. Kadam.
Amruta.
Yes, sir.
Yeah, please. Can you share your live feedback?
Yeah, sure.
So,
Turn your video and go ahead, please. Yes.
sir, excuse
Sanika,
me.
yes. Yes, Sanika. Yes.
Sir, I have doubt.
Yeah.
Okay,
Please,
go ahead.
you can ask.
Go
Yeah,
ahead.
yes, Sanika.
Yes. Sir, there is one post came on WhatsApp that in that, sir, it was there ki by 5G technology people are dying. Some post like that. So, sir, it was true or not?
No, no. There are some evidences.
Yes, sir, in that electromagnetic radiations.
No. There are people are apprehensive about 5G technology because still 4G technology was available. All those towers were far away. Now with the 5G technology, we require multiple towers. And, people are apprehensive. Because there was one paper which was blocked where increased incidence of brain tumors in people, say, residing close to the towers, but then it was not validated. So I think 5G is a new technology. So we need to monitor them and we need to find out how it can be done. But apprehensions are, I think, appropriate.
Yes.
Okay, sir. Thank you, sir.
Yes. Yes, Bobade, sir, you can go with the Amruta T. Kadam, please.
Yes, Amruta is there. Please go ahead, Amruta.
Hello, sir. Good morning, everyone. I am Amruta Kadam. I am Pharm D student and completed internship from Government Medical College and Hospital, Aurangabad. Nitin Jamkar sir has given us a very informative topic today, and I am very happy for the eye doctor chair, as I did my internship in government setup in a tertiary care hospital. I have seen many patients there. When there were many patients, so we cannot manage all these patients. So this will really helpful in the future. And artificial intelligence is very helpful for improvement in the healthcare system, which will save our time and patient will have a quality of life. Thank you, sir. Thank you, MIT team.
Thank you, Amrita. Because medical education technology says you cannot concentrate beyond 45 minutes. So that's why I didn't want to talk much, because this one of the use is in high volume OPD.
Yes, sir.
And high volume OPD, you go there, you just sit in front of machine, and then machine will, we can put 10 machines in Government Medical College, Aurangabad OPD. And then it can be directly seen on the monitor of the resident, and he will reduce the time. So high volume OPD, specialist OPD, all these algorithms can be used. Incidentally, I'm graduate and postgraduate from Government Medical College Hospital, Aurangabad. And
Yes, sir.
I want to talk about big cell therapy and xenogenic transfer factor, which was done in GMC Aurangabad. So I think we all the time say that we don't have ecosystem, But then GMC Aurangabad gave me entire ecosystem for conducting cell therapy, that too in 1975 to 77. So I have a lot of regard and respect for my own college, and that I am proud of my college. Thank you.
Yes, sir. Nitin Naik is there. Nitin Naik, please, can you give your feedback?
So with new idea, that is a finishing school, this workshop.
You hold the mic properly so you can hear. We could hear properly. Yeah.
You're not clearly audible, Nitin. Please go ahead.
Yeah, please.
Okay, sir. Okay, such a new idea, that is finishing school. With this new idea, this workshop is going on with last over 2 weeks. I found that the workshop was very informative for all of us. Today's presenter, Dr. Jamkar sir, was very knowledgeable person in the area of artificial intelligence, and also he has explained us the recent developments in the discovery. So thank you very much, sir, from Shivaji University, as well as from I also thank you for the MIT World Peace University for organizing such an very wonderful workshop for all of us students. Thank you once again, sir.
Thank you, Nitin Naik. So with this, I am concluding.
I think we will take one feedback from our MIT student. Kushagra,
okay.
Then we can continue, sir, please. Kushagra is there? Please.
Yes, sir. Am I audible?
Yes. Not audible. Can you hold the mic properly?
If possible, switch on your video, Kushagra.
Yeah.
Please.
Actually, there is some connectivity issue.
Yeah, now audible.
Go ahead.
Okay. So good morning. I think this session was really very informative, and I was completely immersed in it. Clinical trials and clinical things, hospital is one of my favorite subjects. So I was so involved in it that I enjoyed the session a lot. So thank you so much, sir, for this session. Thank you, Polsettivar sir. Thank you, Burbe sir. Thank you so much.
I just want to volunteer that all the students, they can always approach me for all their difficulties in research and development. I gave one lecture in MIT ADD University in Pune about medical imaging, and there was one student who approached me, said, "Sir, I'm working on COVID pneumonia," and then he was working with say TensorFlow. He developed a image handling software, and then I help him by giving say I connected just to another hospital, and he was given say more than 50 X-rays of corona, and then he developed that whole image handling intelligence with reasonably perfect. And then I connected him with one of my company where I'm a mentor called Rises. And then at the end I said, "What are you doing? Are you doing in BTech or BE or what are you doing?" You'll be surprised to know that student was appearing for 12th standard. And then the whole thing he was doing for getting admission through the SAT. So I think we have no limitation of intelligence in students. So hats off to him. I just forget his name because anyway. So I would request all of you to think original, think innovative, and just don't stop at small things. Go on doing, go on reading, doing review, and do innovations and startups. And whole Indian ecosystem is promoting innovations, promoting industry, promoting startups. So you just want a few ideas. Now the simple ideas. You go to the hospital, and then there is one patient who is receiving IV. And then if you look at them till the bottle is finished, several people just watch and find out bottle finished, bottle. Now you require a simple thing. You can put maybe a weight sensitive, say IoT, where 500 grams, the moment it is 10 grams, it can send a message. So it doesn't require a rocket science. But then if you develop something like this for monitoring an IV fluid, it will sell like a cake, because every hospital, they have minimum 1,000 IV sets. So I think we need to look into this direction, that medical technology can be used for a better healthcare. Thank you.
Thank you so much, sir. I think I really appreciate your efforts, and last two sentences was really a cream and carry home message for all of us. Sir, I take your opportunity to say a tagline for Dr. Arun Jamkar, that think original, initiate innovation. From this to
Sir, we lost you. Maybe some connection issue is there. Bobade sir, we lost you.
Dr. Satish, how many people were watching on YouTube?
YouTube sir, near about 60, say 70, and here 43.
Okay.
So now it is more than 100, sir. If now, because I have shared the link to many students, now it is cross 150. Initially it was 7, now 160 above.
Yeah. I'm there,
Yeah,
sir.
Chandrashekhar sir, we lost you. Can you just repeat?
Yeah, sir. In between, it's unstable. So I'm here to conclude the today's session. This was really definitely a great ecosystem healthcare session, and sir has not connected the disciplines, but sir has connected the many people together from the different discipline. I have witnessed one example of regulatory affairs that sir that much of hunger to connect the interdisciplinary people together and to build the science. So we have discussed machine learning, precision medicine, personalized medicine. Artificial intelligence will be a booster in the medical treatments, immunocancer therapy, genomic contents, then circulating tumor cells we call as the CTC, which example of our own monoclonal antibodies. So what are umbrella clinical trials? What are the basket clinical trials? And monitoring the clinical trial with the help of IoT. Then IoT in healthcare, a role of stimulation techniques in a medical history, and last, a doctor has created an eye doctor. This is a great example of sir's contribution to upgrade the medical ecosystem. And sir has spoke about he's a persistent with the persistence, and because of that he could invent the eye doctor, which will serve the healthcare need of the people. Sir, we are very much thankful on behalf of School of Pharmacy, on MIT-WPU University, and our own Dean, Dr. Nanesh Lime, who is a true initiator of this finishing school, and all we colleagues are basically carrying it and taking with the help of you, exemplary people in the field, and we are taking to the student. So with this, I extend my sincere thanks to all the stakeholders, all the leaders of the MIT-WPU University, our own Dean, and on behalf of that, I request and I extend my sincere thanks to Dr. Arun Jamkar sir, and we would like to hear from you in the future as well. So with this,
we'll take good photo.
I request all switch on your cameras. We will capture this incident, this moment of great talk, and to cherish the memories. So I request all the audience and delegates to please switch on your cameras.
Students, put on your camera. 1, 2, 3. Thank you.
Please, all.
Again, I'm taking.
All of you, please put on your cameras on.
Thank you.
Thank you. So with this, we can the session. Satish sir.
Yes, sir. Yes, sir. You can now conclude the session. Thank you, sir. Jamkar sir. Really, we are very fortunate.
Thank you. So we can say the session is over. Thank you, sir. Thank you so much.
Thank you, sir. Thank you for your great words and
thank you.
We look forward to your cooperation, sir, for our student project.
I'll just conclude with one statement. Let us work together for a nation building activity to make this country great.
Yes, sir. Thank you, sir.
Yes, sir. Thank you, sir.
Thank you.
So conclude the session.
Yes, sir. Yes, sir. I'm closing the session.